Hi, welcome all. Welcome to Free License class. So, today we are going to study about lac operon. Okay, so what is operon? Operon means it is a combination of genes like regulatory genes and structural genes, promoter operons, which will help in regulating the gene expression. Okay, so the purpose of the regulation of gene expression in prokaryote are remarkably well served by the use of operons. Okay, so in prokaryotes, the operon is very essential to control the gene expression. To control the gene expression. So note that it is very important for the to regulate the gene expression. Okay, all the genes of an operons are coordinately expressed and the metabolic pathway controlled by this operon can be regulated very fast. Okay, my network have some issues, so there is some delay. Okay, okay, I hope everything will be okay. Okay, let's start our class. So all the genes of operons are coordinately expressed and the metabolic pathways are controlled by the operon can be regulated very fast because every operon have more than 3 or 4 genes. So what happens is that when you regulate one single gene, you can control 3 different structural genes. For example, in lactose operon, you can control 5 different structural genes. For example, that in the case of tryptophan. Likewise, there are more than one gene so that it is easy to control more than one genes in single way. Okay, in single controlling a single promoter. And the energy saving as the same set of regulatory sequences and the promoter is used for all structural genes of the operon. That means you are saving a lots of energy because energy is very essential. So when you use energy, that is when your bacteria, the E. coli, if use energy, they need a lots of energy for the lactose metabolism. Okay, so when this sorry lactose metabolism, so when they use energy to regulate one promoter, you can control 3 structural genes. So the energy used to transcribe the 3 different genes are same. That is, here you are saving more energy. So when comparing the metabolic pathways in prokaryote or in any organism, you can see there are 2 different pathways in metabolic pathway. That is, one is catabolic pathway and the other one is anabolic pathway. So in catabolic pathway, you can see the breakdown. Okay, the breakdown of macromolecules. And in the case of anabolic pathway, you can see the synthesis. So these 2 pathways are entirely different. One is breakdown, and the other one is synthesis. So one of the most important challenges that is faced by the prokaryote is the transition from one metabolic pathway to another. Because if your environment is changing, okay, if your environment is changing, what is the result? If the environment is changing, that means you need to change into some other metabolic pathway. That means you have to change, you have to spend more energy. So you need to change the gene to on position and the first gene into off position. If you compare it with, or if I say with an example, the bacteria is using glucose metabolism first, and if the environment is changed, that is glucose is consumed and there is no glucose at all. So they have to change from glucose metabolism to any other substrate like lactose or any other sugar which is present in the source, that is environment. So you have to change from glucose metabolism to lactose metabolism. You have to switch on the gene. So here comes the um, significance of operon concept. Here you change the gene of lactose to on position, that is lac operon get on. Okay, so in catabolic pathway you need a substrate here, that is lactose. So you have to convert the lactose into smaller units, that is to convert into glucose and galactose. You need to switch on the gene. So in the operon, you just switch on that gene, that is the lactose metabolism will takes place. In anabolic pathway, what will be there here? The product which the product is already present in the cell, that is your glucose is always there. So you need not want to convert lactose into glucose and galactose by spending lots of energy, so that you will make the gene switched off or remain that gene as off position. So this is one of the repressible operon. Okay, so if we compare all the thing in gene regulation, we can say that gene can be regulated by different means. As we have said earlier, we can control the gene expression, okay, according to environmental condition. We can control by environmental condition. Likewise, if the physiological condition is changing, okay, physiological contagion is changing, then also you regulate your gene expressions. Likewise, if your metabolic pathways, okay, metabolites or metabolic conditions are changing, then also you will control your gene expressions. And in prokaryotes, all the process like RNA processing, translation, transcription, everything is happening in the cytoplasm. And the most interesting thing is these all processes are interrelated. That is, if from the DNA, if the mRNA is synthesizing, at the same time this mRNA is converted into proteins. Okay, so these processes are interrelated, that is from DNA to mRNA to proteins, all are interrelated and all are taking place in the cytoplasm. Hence in most prokaryotic cases, okay, in most prokaryotes, their control is done at transcriptional level. Okay, most of the cases it is done at the transcriptional level, control at transcriptional level. So operon is one of the example for transcriptional level control. Okay, in the we are in this video we will discuss only about lactose operon. In the next class we will discuss about tryptophan and as well as arabinose operon. So what about lactose operon? So here you can see a graph. Okay, here in this graph it is shown that growth of bacteria. Okay, here it is given that E. coli is grown in the medium which contain glucose as well as lactose. So what happens when there is glucose? They utilize glucose and they start to grow. When the glucose is consumed, okay, totally consumed or fully consumed, what happens? The bacteria will stop growing and they go into a stationary phase. Okay, they grow into a stationary phase. Okay, here there is no growth, they move into a stationary phase. Okay, they get a transition from growing phase to stationary phase. At that period they will change their metabolic pathway. They will change their metabolic pathway from glucose metabolism and they change it into lactose metabolism, because there is the presence of lactose in your medium. So they change their metabolic pathway. So when the lactose metabolism is started, again they start growing, and after totally the lactose is consumed, again they go into a stationary phase or a plateau phase. If there is no glucose and lactose in the medium, after some time you can see a decline phase also, that is death phase or decline phase of the bacteria. If you give another metabolite like arabinose, they will choose for arabinose metabolism. Then again you can see one more log phase and one stationary phase, that is stage 3. You can also see stage 3 growth. So what is the meaning of these two stages? The first stage is when the glucose is present, which is their primary carbon source. Okay, primary carbon source, instant energy, and this glucose is more preferentially adapted in the bacterial system. That is, this glucose metabolism is as well adapted in the bacterial system. Hence, they use the glucose as the primary carbon source. So they adapt the glucose metabolism in the first stage. After it is expired, they will go for your lactose. Okay. Why this second phase? That is question number 3. What does this mean? What does this mean? It means that is the transition period from you are changing from the glucose metabolism into lactose metabolism. That is, you are inducing the expression of lac operon. You are putting on the gene of lac operon. That is happening in this stage 1 plateau phase. Okay. This is actually what we are going to discuss in this video, the lac operon. Here you can see the beautiful diagram of a lac operon. So in this structure of lac operon, you can see structural genes, that is gene Z, gene Y, and gene A. So 1, 2, 3. You can see 3 structural genes in the lac operon. Okay, 3 structural genes are present in the lac operon, and these structural genes are controlled by this promoter, and nearby there is an operator site where the effector molecules come and bind. So what is effector molecule? Effector molecule is nothing but it is a repressor or activator. Okay, either it can be a repressor or it can be an activator. Next thing is about the gene I. So this gene I is very essential to make the repressor molecule, the effector molecule. Okay, this effector repressor molecule will bind to the operator if there is no lactose in the medium and make this operon off. Okay. And this promoter for gene I, this is the promoter for gene I, which helps in regulating the gene I. Okay. And this gene Z will produce a protein known as an enzyme known as beta galactosidase, which helps in cleaving the lactose into glucose and galactose. And the next gene is gene Y, which helps, this mRNA will produce another enzyme called beta galactosidase permease, which helps in pumping the lactose into the cell. And the next enzyme is transacetylase, which is produced by the gene A, which will help in eliminating the toxic thiogalactosidases and also transported by the lac Y into the cell. Okay, these are the functions of enzymes in the lac operon. Next, we are going to discuss more about lac operon. So this lac operon, so we have already studied about the structure. Once again, I am saying that this operon is basically composed of a promoter, operator, and structural gene, and also it have regulatory gene. Okay, so the lac operon have promoter, operator, structural genes, and regulatory gene. So the lac operon is a negative inducible operon. Okay, this lac operon is a negatively inducible operon. Negative inducible operon means it can be um, switched off by using repressor, and it can be switched on using activator. So what is inducible operon and what is repressible operon? I will discuss that. So first of all, I will write like this: positive control and negative control. Okay, positive control as well as negative control. There are two types of control in lac operon. In positive control, that their lactose is present. Okay, so when lactose is present, allolactose, which is an isomer of the lactose, will come and bind with the repressor molecule. Okay, so the repressor molecule will change its conformation. Okay, will change its conformation and dissociate from the operator site, so that RNA polymerase comes and bind to that site. So what happens? Transcription will take place. So by the attachment of this lactose, this effector molecule to the operator site, you have changed the gene position into on. That is, you have switched on that gene. Such type of control is known as positive control, and you can also say that it is a type of inducible operon, because this operon is changed on or switched on by an inducer or effector or an activator. So this is known as inducible operon. In the same case, if the lactose is absent, okay, lactose is absent, then what happens? There is no activator molecule, that is, there is no lactose. So the repressor, the gene I will produce the repressor molecule. Okay, the gene I will produce the repressor molecule, which in turn bind to the oocyte, which will block or inhibit the transcription. Okay, that is the thing will happen in the negative control. So you can see both this negative control as well as the positive control in the lac operon. Okay, so the lac operon have regulatory sequences. Okay, the regulatory sequences are the operator site and the promoter site. So the first thing is the operator site, which binds to the repressor protein, and the second one is the promoter site, containing two binding sites, one for the RNA polymerase and other for CAP's cyclic AMP complex. I will discuss it later. Okay. Next, about the structural genes. The second thing in the lac operon is the structural genes. We have already discussed about that. That is gene Y, gene Z, gene Y and gene A. And about the regulator gene is the gene I, which code for the repressor molecule, and one promoter for the control of this gene I are regulator genes in the lac operon. Okay, so for saving energy, the lac operon is switched off in the lactose-free media. Under this condition, a small amount of lac repressor is produced. Okay, when the lactose is absent, lactose repressor will bind to the operator region, which overlap the promoter. So likewise, here you can see this is the operator site and this is the promoter site. When the repressor command binds to the operator site, it will overlap this promoter, so that RNA polymerase cannot see the promoter and will not bind to the promoter. So their transcription will not take place. Even though there is a weak activity is observed, and a small amount of these enzymes are produced even the lactose is not present in the medium. But if it is not the case of lactose is present, if the lactose is present, this lactose comes and binds to this repressor molecule, so that a conformational change will occur. Okay, this conformational change will occur and it will detach from the operator site. So this will increase the binding of RNA polymerase, okay, RNA polymerase to the promoter. At the same time, there is another site in the promoter where the CAP cyclic AMP will bind to this promoter and will promote the transcription. Okay, so what is catabolic repression? So during glucose metabolism, okay, during glucose metabolism, you have to inhibit the lactose metabolism. For that purpose, the glucose molecule, the higher concentration of this glucose will inhibit another enzyme known as adenyl cyclase. Okay, it will inhibit another enzyme known as adenylate cyclase or adenyl cyclase. So when this enzyme is inhibited, it will not produce cyclic AMP from ATP. That is, the function of this adenyl cyclase is to form cyclic AMP from ATP. So when this enzyme is inhibited, it cannot convert the ATP into cyclic AMP. So what happens? There is no production of cyclic AMP, and this process is known as catabolic repression or glucose effect. That is, inhibition of this adenylate cyclase is known as catabolic repression or glucose effect. Okay, so this is more about catabolic repression. So now glucose is present in higher concentration so that our adenyl cyclase is inhibited. So there is no conversion of cyclic AMP. But if the glucose concentration is reduced or the glucose is fully expired, what will happen? We have to switch on to lactose metabolism. So when this glucose is reduced, it cannot inhibit adenyl cyclase. So this adenyl cyclase get activated. It will suddenly convert ATP into cyclic AMP. So here you need a lots of energy, like because to produce cyclic AMP, ATP to cyclic AMP, lots of energy is spent here. Okay, so when more amount of cyclic AMP is produced, this cyclic AMP then will bind to another protein called catabolic activator protein. So this will associate with this catabolic activator protein and will form a complex. This then went and bind with the promoter. So it will enhance the RNA polymerase to bind to the promoter site. Okay. So this is the need for cyclic AMP in the lac operon, because the RNA polymerase, whether the promoter is not strong, the RNA polymerase cannot come and bind to the promoter. So there is a consensus sequence present in this structural gene, that is a 35 sequence apart, that is upstream to that promoter. So this will not favor the RNA polymerase to bind. So RNA, to make this promoter strong, we need another molecules. Here comes the significance of this CAP cyclic AMP complex. So this catabolic activator protein cyclic AMP complex will help in binding of the RNA polymerase, which in turn induce the expression of lac operon. Okay, this is all about the lac operon. I hope you all understand about lac operon. If you liked my video, please give me a thumbs up. And if you don't like this video, please give me a dislike so I can improve my video. So please give me a subscribe. Please subscribe to my channel and click on the bell icon so that you will get notification when I upload more videos. So I will be uploading more and more videos so that you will get notification only when you click on the bell icon. So thanks for watching my video. Love you all.